As you see from the title here, Olivia and I will be talking about supposedly benign biological interventions to reduce offending, because if retributivism is to go, we really need treatments that work. I think that's going to be critical. So to sketch out what we're going to talk about, I'll start with some basic science research on the amygdala and psychopathy. I'll look at the brain in prison. What happens to the brain when it's in prison? Not so benign. We'll turn to research on castration. I'll turn to research on drugs, medication, which again, many people would not think are benign interventions, and probably they're not. But we will then turn to other interventions like nutrition, biological interventions to change risk factors for crime. We'll turn then into looking at one risk factor for crime, prefrontal dysfunction, and then what we could do about it. Could we use transcranial direct current stimulation, for example, to reduce criminal intent? And lastly, we'll look at a brain area called the striatum, which is very much involved in the reward system. In terms of the layout of this, I'll talk about the first part. Olivia will come in and talk on the second part. And just as a word of caution, we are not lawyers, we are not philosophers. So we're just basically empirical scientists. How any findings are interpreted are largely up to yourselves and your impressions. We'd appreciate very much. Well, let me start with just the amygdala and psychopathy. So I'll talk about some basic science here. You'll be familiar with psychopaths, those individuals who lack conscience, they lack remorse, they lack guilt, they don't experience the emotions that we do. The question, why is that? Is that because they decide to be immoral, or is there a neurobiological substrate underlying this pathology of psychopathology? What we found in comparing 27 psychopaths with 32 non-psychopaths is that there's a shrinkage in the amygdala. The amygdala is the emotion center of the brain, very crudely speaking, lying roughly here deep inside the cortex. Overall, we found that the psychopaths had an 18% shrinkage in the volume of the amygdala. So the key science point from this is the reason why psychopaths act immorally or lack conscience, lack feelings, is possibly because of deformations in the amygdala, and blue indicates a specific area of the amygdala that is structurally deformed in psychopaths. That's brain imaging research. Problem with brain imaging research is that it's cross-sectional. What's causing what? Does a reduction in the volume of the amygdala in some way predispose to later psychopathic behavior? Or is it the other way around? Could living a psychopathic way of life change the brain? Because we know the social environment just does change the brain. We don't have longitudinal research assessing the amygdala early on in life, but we can come fairly close to that by measuring a construct called fear conditioning. Another term is classical conditioning. Do you remember Pavlov's dogs? It's an emotional form of learning. And the important thing here is that fear conditioning is a proxy for amygdala functioning. All neuroscientists would say, yes, where does fear conditioning come from? It comes from nuclei within the amygdala. So we had an opportunity to try and tease out the temporal ordering of the variables in this study in Mauritius. I'll just describe it briefly here. We have 1,800 3-year-old children who are assessed on fear conditioning at age 3. We measure fear conditioning from having electrodes placed on the finger to measure sweat rate. I'm not going to go into the paradigm, but take high fear conditioning as a measure of fearfulness and a measure of amygdala functioning. Of those 1,800 3-year-old children, they were followed up for 20 years, and 137 of them had a criminal conviction, caught and convicted for a significant crime. They were matched with controls on gender ethnicity and 11 indicators of social adversity. And the key finding is this, that if we go back in time 20 years, from age 23 to age 3, and look at fear conditioning, this is good fear conditioning, this is poor fear conditioning. The green bars are the little 3-year-olds who grow up to become criminal offenders. Blue bars are the 3-year-olds who do not grow up to become criminal offenders. And what we find is that poor fear conditioning, think of poor amygdala functioning at age 3, is a risk factor for future criminal offending. It's not perfect prediction, of course. This is an effect size. This is Cohen's d. If you're not familiar with Cohen's d, 0.2 is small, 0.5 is medium, 0.8 is large. Small effect sizes. Of course, there's more things to committing crime than poor amygdala functioning. But we are teasing out, to some extent, the temporal ordering of the variables. We think that poor amygdala functioning comes before criminal antisocial offending. Okay, when this was published, the journal who published it, American Journal of Psychiatry, ran an editorial, a critique written by a basic scientist called Philippe Sturzer. This is the title, Born to be Criminal. You know, what do we make of this? And Sturzer says, look, we've got to be very cautious here about using neurobiological markers to mark out who is going to become criminal. That could be stigmatizing, and I think we would all agree there. But Sturzer goes on to say, but we're gaining new insights into the etiology, the causes of crime. And Sturzer's last sentence is, so we should use this to benefit those children who are greatest needs, and let's create interventions to cater for them. And I think we could look at this from two perspectives. On one perspective, Greg, public health approach, screen and intervene early on, prevent crime, stop it before it starts, you don't have any more retribution. That's one side of the equation. The other side of the equation is the potential for stigmatization and marking out children early on in life on the basis of biological risk factors as would-be criminals. I think that's an area that a section of society would not want to tread. But that's open for debate and discussion. We'd be interested in your thoughts on this. Staying briefly with the amygdala, the other thing you need to know about the amygdala is it's replicated in many labs that the amygdala is quite central for moral decision making. Brain imaging research shows that when we make moral decisions, the amygdala is quite strongly activated. Andrea Glenn, one of my graduate students, did a study on moral decision making in psychopaths and non-psychopathic individuals. Greg and I were talking about this. It's the good old trolley dilemma, Greg. But this is just one example of a number of moral dilemmas that were given to the participants. And if you're not familiar with the trolley dilemma, it's very simple. You're standing on a footbridge next to a large gentleman. Runaway trolley is going to kill 5 railway workers. Do you push the big man off the bridge because he stops the train? You save 5 lives, but of course he dies. Utilitarian moral decision-making or not. So, Andrea brain scanned a number of individuals who varied in their degree of psychopathy. So every red dot is a person. This is psychopathy score on the x-axis. On the y-axis, we have amygdala activation, high and low. She found a correlation. Small sample size, by the way, we must be careful. But the direction of effects is the more psychopathic the individual is, the lower the amygdala functioning when they're making personal moral decisions. So this to us gives rise to a question, and this gets into more the legal arena, potentially, I think, and it's this. Of course, psychopaths cognitively know right from wrong. They know it's wrong to kill. No question about that. But the question that last study perhaps raises is, do psychopaths have the feeling, the emotional feeling for what is right and wrong? Because that imaging study suggests they do not, especially in a moral context. And we believe that emotion is part of the engine that drives moral thinking and moral behavior. And if the amygdala, the emotional engine that drives moral thinking, if that amygdala is burnt out, then how, and if psychopaths are not responsible for having a burnt out amygdala, then how responsible is it of us in society to punish psychopaths as harshly as we do? Because I don't think psychopaths ask to have an amygdala that's, 3 sizes too small. When I worked with prisoners for 4 years and I asked prisoners what they wanted to be, when they were kids, how they wanted to grow up, they said, well, I want to be a football player, I want to be a racing car driver. No criminal ever said to me, I wanted to grow up to become a psychopath. Where is free will there? Okay, I want to turn now to the brain in prison. Very surprisingly, the brain in prison, we know very little about the brain in prison, meaning we don't know what happens to the brain when it's in prison. What happens to cognitive functioning? Surprisingly, there's been a lot of speculation that prison is not good. Surprisingly, no data. It's really very surprising. So I'll describe to you some study. Yes, Greg, Rikers Island. You've heard about Rikers Island before. My graduate student, Becky Umbach, was involved in a study where she could collect data on young offenders when they go into Rikers Island, and then 4 months later. What does living in Rikers Island in the prison institution do to the brain? Let me describe the cognitive task. It's called the emotional go, no-go task. To give you an example of this task, it's a measure of emotion regulation and impulse control. So, you're shown different faces with different emotions. You've got a response button. You press the button when you see a neutral face. You do not press the response button when you see an emotional face. So you see a face like this, it's neutral, so you go, you press the response button. You see this face, neutral, you go, you press the response button. You see this face, you don't go, because the person is smiling there. So that's no go. See this face, neutral, you go. So that's the task. What happens in prison over 4 months? So just look at the top of the slide here. This is response inhibition. You need to know that a high score indicates poor performance. That's how you read this graph. This is the prisoners when they start in prison at 0 months, and this is giving the same task to them 4 months later. They get worse. High scores are poorer performance. So in terms of cognitive functioning, they do worse after 4 months. The effect size is small, but of course they've only been in prison 4 months, and they're not in total isolation, for example. They've got the run of the prison. This is the emotion regulation score. It's significant, a significant decline in prison. So surprisingly, we've ignored what happens to the brain in prison. But what it's doing, Greg, it's taking a risk factor for crime, because lack of impulse control is a risk factor, emotion regulation is a risk factor. We put them in prison, we make the risk factor worse. That's why they come back. Now, what I haven't told you is that actually this is the average of 2 groups. Because actually this is embedded in a treatment study, because this talk is about treatment. Half of the prisoners went into a mindfulness training and cognitive behavior therapy intervention. The control group had an active intervention. It was drug abuse awareness. And so let's look at these data when we split up the 2 groups. What we find here, if you look at the red group, they are the control group, and there's a significant effect size. They significantly get worse in time on the task. But if you went into the mindfulness or cognitive behavior therapy intervention, it's more of a flat line. Actually, there's no significant decrement in imprisonment. And that's true also for emotional regulation. So on both counts, mindfulness and cognitive behavior therapy appear to protect you from the negative effects of imprisonment. And so would we think of a benign intervention like mindfulness and cognitive behavior therapy for offenders in prison? Okay, not so benign, castration. But let's look at it. Castration in sex offenders. I was always intrigued by this early study from the Federal Republic of Germany when castration in prison was voluntary. An intriguing study of 100 castrated sex offenders, 35 sex offenders who wanted to get castrated and they never did. And they did an 11-year follow-up after release from prison. And I want to draw your attention to sex offending 11 years afterwards. In the controls, it was 46%. In the castrated group, it was 3%. So no matter how heinous castration is, it does reduce sex offending. That's physical castration. 10 more studies in these countries without a control group. That's a problem. There's no good scientific work in this group. low rates of sex offending. And a study of 2,000 castrates from Europe, sex offending post-release between 0 and 7%. So no matter how barbaric it is, my question is, would we at least give sex offenders the option for castration? And what about chemical castration, which is more reversible? Again, no good control studies, but a number of investigators arguing it is effective in reducing sex offending, and certainly it's mandatory in California and Florida for some types of sex offenders. The question again I have for you, no matter what your thoughts of this as being barbaric, would we give the option to prisoners to have this in lieu of imprisonment? Would we allow them to decide for themselves or not? What about medications? We never think about medicating prisoners. I wonder if we should think about it, because what we know, certainly, without any question whatsoever, medications are effective, very effective, in reducing aggression in certainly children. Randomized controlled trials, medium effect size, and a whole variety of medications do reduce aggression in children. There's no question about that. Amazing study on adults by Lichtenstein in the New England Journal of Medicine came out. Very large sample of ADHD patients, 25,000. What they showed is that giving stimulants, amphetamines, to offenders, both men and women, it reduced criminal offending in men by 32%, in women by 41%. So would we do that? Would we think about that type of biological intervention or not? Nutrition. How about a natural form of medication? One example is omega-3, a long-chain fatty acid, critical for brain structure and brain function. We did a randomized controlled trial on the island of Mauritius here. We give children, 200 children, randomized into two groups. 100 get a fruit juice drink every day containing 1 gram of omega-3. The control group are getting the same fruit juice drink, but it does not contain omega-3. These are the findings: child antisocial behavior, high and low, 0 months, 6 months. This is the treatment duration. 12 months is a 6-month follow-up. Two things happen. Both groups come down. That's a placebo effect. That happens in treatment studies. People think they're getting an intervention, they do better. But you see the red line, the controls, when the treatment ends, they begin to go back to where they came from, whereas the omega-3 group continue to decline. Again, a small to medium effect size, not enormous. That's not going to, stop crime, but is it something that we would think about? Very few studies on prisoners. We're doing a study in Singapore at the moment here, where the omega-3 group are beginning to show a decline in antisocial personality compared to these two red and blue control groups. So bless you. So in the experimental group, there's a 66% significant reduction in antisocial behavior, at least after 3 months. But what about the long term? We don't know that yet. And just very lastly here, just really to lead into Olivia's part of the talk, what we also know is that omega-3 does upregulate the prefrontal cortex. We won't go into any of the details of that study by McNamara, but this part of the brain, the dorsolateral prefrontal cortex, is being upregulated in at least young boys. With that being said, it's over to Olivia.
Okay, so the finding of the effect of DHA supplementation on frontal functioning is particularly relevant and significant when we think about, when we consider the prefrontal dysfunction that we see in antisocial individuals. So earlier we talked about the amygdala and deficits in the amygdala that have been found in, for example, in psychopaths versus controls. So we'll turn our discussion here back to the brain, albeit a different region of the brain. So in an early imaging study that investigated the type of brain dysfunction that may predispose to violence, the brain functioning in murderers who pled not guilty by reason of insanity, so these are violent offenders, were compared to the brain functioning in a sample of controls using PET scans. So essentially, a tracer is injected into a participant that's taken up by the brain as a tracer of brain metabolic rate. And so what the study found, what the study led by Adrian found, was that the group of murderers, 41 murders, showed lower glucose metabolism in the prefrontal cortical areas compared to matched controls. In other words, we see these deficits in the prefrontal cortex that we don't see in offenders, as you can see with the difference there between these two different scans. So since then, studies adopting other neuroimaging techniques as well have found that prefrontal brain impairments are one of the best replicated risk factors for antisocial behavior. So a meta-analysis that included all prefrontal and prefrontal subregional findings indicated that antisocial individuals show both reduced structure as well as function in the prefrontal cortex with a moderate effect size, as you can see up there. And more recently, this has extended to sexual offending as well, with the finding that rapists show decreased conductivity in white matter near the medial frontal pole compared to matched controls. All right, so more specifically, one subregion that has been particularly implicated in antisocial behavior is the dorsolateral prefrontal cortex. So it's that area you see there in the picture. And on the right, you can see the right and left dorsolateral prefrontal cortex bilaterally. And it's been suggested that this may be due to this region's broad connection to functions related to violence. For example, there's evidence that the DLPFC is associated with psychological risk factors for antisocial behavior, like moral judgment, for example. All right, so since this talk is on treatments, on interventions, and given that prefrontal deficits is a risk factor for antisocial behavior, well, one way of maybe targeting these deficits is through an experimental form of non-invasive brain stimulation called transcranial direct current stimulation, or TDCS. So what TDCS is, as I mentioned, a non-invasive technique through which a continuous, low-intensity current, so it's very weak, is delivered through the scalp from an anodal electrode to a cathodal electrode. So this is an example of a TDCS device, as you can see there. It's connected to two electrodes, so one would be an anode, the other would be a cathode. Anodal stimulation increases, it excites or increases neuronal activity, whereas cathodal stimulation reduces or inhibits neuronal activity. And so that way, theoretically, you can either upregulate or downregulate activity in a brain region. So in the past decade, there really has been an increase in the number of TDCS studies, but only quite recently have a few studies begun to apply TDCS to the study of aggression. And these studies have specifically looked at the effect of TDCS on a specific lab task called the Taylor aggression paradigm. But there have been some really mixed findings here. For example, one study found that when participants were given insulting feedback and they were angered, after that, when they received upregulation to one side of the dorsolateral prefrontal cortex, that these individuals who received the stimulation behaved more aggressively when they were angered. On the other hand, another study found that when participants received upregulation to another side of the dorsolateral prefrontal cortex, that they showed reduced proactive aggression specifically, and only in males. And so, there's a limited body of literature here, and also the findings seem to be really mixed. But, these are all based on one specific lab task. What about criminal intent, intentions to commit violence? So this is what we looked at in a double-blind, placebo-controlled, stratified, randomized trial in Philadelphia, in which we recruited 81 adults from the community. And so these individuals were brought into the lab, and they were randomly assigned into one of two conditions. So they were either assigned to an active TDCS condition, so they received stimulation, or to a sham condition, and this is just a placebo. So regardless of which condition they were in, participants received two electrodes, two anodal electrodes. So again, anodal theoretically upregulates activity to the dorsolateral prefrontal cortex bilaterally. So as you can see in the picture there, the right and left prefrontal cortices. So this happened for both groups, and a cathodal return electrode was placed at the back of the neck. For participants in the active stimulation condition, they received 2 milliamps of current to the prefrontal cortex, so 1 milliamp to each of those sites. And they received the stimulation continuously for 20 minutes. For individuals in the sham condition, they only received stimulation for 30 seconds. And this method has been shown to be effective for blinding, so participants don't really know which condition they're in. All right, so in addition to a number of baseline measures that we assessed prior to the stimulation, just to give you a brief idea, some of these traits included sort of baseline levels of offending, baseline aggression, psychopathy, impulsivity, for example. 1 day after stimulation, hypothetical vignettes were given to these participants to assess behavioral intentions to commit violent acts. So we looked at 2 specific types of violent crimes, a physical assault and a sexual assault. So basically what happens is respondents are given this hypothetical vignette in which someone commits a certain violent act, and then they're asked to respond or rate the likelihood that they would engage in the act as the protagonist does in the scenario. So just to give you an example, this is the physical assault scenario. Basically, at the end, someone hits someone else over in the head with a weapon. Well, in this case, with a bottle. So then participants rate how likely it would be that they would engage in this violent act. All right. So when we look at group differences, the differences between these 2 groups on the outcome measure, what we find is that there's a main treatment effect on here the overall likelihood that they would commit any sort of violent act. So on the y-axis here, we have sort of the likelihood that they would engage, that they report that they would engage in that violent act. The red bar represents the TDCS group, the group that received active stimulation. The blue bar represents the placebo group, the sham group. And what we see is that the individuals who received active stimulation reported a lower likelihood of engaging in the aggressive act in the scenarios. If we look at physical assault and sexual assault specifically, we see a similar pattern of findings there, where individuals who receive active stimulation report a lower likelihood that they would behave as the protagonist does in those scenarios compared to the controls. All right, so beyond that though, what accounts for that reduction in intentions to commit crime? So in addition to asking participants to rate the likelihood that they would engage in those acts, we also asked them to rate on a scale from 0 to 10 how morally wrong they perceive the act to be. So we're looking here at perceptions of moral wrongfulness. And what we found was that looking at those 2 groups, the y-axis here, you see ratings of moral wrongfulness. So a higher score means that participants rated those acts as more morally wrong. That individuals in the active stimulation group reported enhanced perceptions of moral wrongfulness. So they rated those acts as more morally wrong compared to the sham group, with a small to moderate effect size here. So if you look at this mediation model, what we find here, testing this model, is that perceptions of moral wrongfulness partly mediated the reductions in intentions to commit those violent acts that were brought about by the active stimulation. So in other words, TDCS influences perceptions of moral judgment, at least to a certain extent, which in turn is associated with the reported likelihood of engaging in those violent acts. And it seems 31% of the effect of TDCS on overall violent intent was accounted for by enhanced perceptions of moral wrongfulness. All right, so a year ago, this article was published in the American Journal of Bioethics, arguing that deep brain stimulation is, it's unethical for psychopathic prisoners. And in an article in response to that Target article, we agreed that yes, deep brain stimulation does not conform to ethical guidelines, although not necessarily for the reasons the authors state, but because of its invasiveness, it's not minimal risk, and given the extant research we have on this, there's no evidence for its efficacy that would justify this. But could something like transcranial direct current stimulation, a non-invasive form of brain stimulation, low risk, non-invasive, could it in the future, and granted these are initial findings, it's initial evidence, there's need for application, but could something like this be, in the future, something that could be ethically offered to offenders, given that it's relatively benign, not benign, but, the side effects are transient and it's relatively safe. All right, so moving on from that, compared to the prefrontal cortex, the striatum is a brain region that has received relatively less attention in the literature, but it's increasingly being studied in relation to antisocial behavior, specifically with psychopathy. And the striatum is this subcortical region that you see here that's located quite deep underneath the cortex in the forebrain. And just very briefly, it's implicated in the processing of rewarding experiences. It's also been documented to be associated with impulsivity, which is very relevant to psychopathy, to antisocial behavior more generally. In the first study looking at the relationship between the striatum and psychopathy, what Andrea found was that psychopathic individuals have an enlarged striatum. So there's about a 9.6% increase in the striatum in in psychopathic individuals compared to matched controls. And since then, there have been a few studies with sometimes mixed findings. Some find evidence for an enlarged striatum. Another study on violent offenders with psychopathy found that those individuals had lower volumes of the nucleus accumbens, which makes up part of the ventral striatum, compared to control. So there's sort of mixed findings here, and what we did was look at whether there are structural striatal abnormalities between psychopaths and non-psychopaths in a different sample. So what we did is participants were recruited from temporary agencies in Los Angeles, and their subcortical volumes were assessed from structural magnetic resonance images in 108 males. So very quickly, what we find here is that on the y-axis, we have the total volume of the striatum. The blue bar represents individuals who met the cutoff for psychopathy. The red bar represents non-psychopathic individuals. And what we find is that the striatum is increased in the psychopathic group compared to controls. So it's about an 8.7% increase that we see here. And so these findings support the notion of an enlarged striatum in psychopathic individuals. And it sort of suggests that, these psychopathic individuals may be more sensitive to reward. Enlarged striatum may contribute to some of the distinct features of psychopathy, like more reward-driven, more impulsive behavior. And it may be that these individuals are overly focused on rewards, even when there are signals of later punishment. And this heightened sensitivity to reward may be a motivation for criminal behavior. And so as it pertains to interventions or treatment, it may be that, a focus on rewarding experiences may help to maximize treatment gains. For example, we can think about, in terms of treatment, maybe for these psychopathic individuals, that more reward-based components of interventions, for example, in children, if we think about sort of more praise, more reinforcement, that may be more effective than sort of the disciplinary components of certain training programs, certain psychosocial interventions. All right. So just to give you a brief summary of all that we've talked about, we started off talking about some of the risk factors for antisocial behavior with the amygdala, deficits that we see there, and the brain in prison, and then a few different types of interventions ranging from more invasive to less invasive ones to ones we would even consider sort of more benign, like nutrition, for example. And we also went on into some brain-based intervention techniques that may be non-invasive. And then lastly, striatum and the enlargement in the striatum and how that can inform how we approach interventions with our knowledge on how psychopaths perceive rewards. And so we just wanted to end the talk with a few questions that we think are relevant to our findings, and that we hope will stimulate some discussion and also your thoughts on this topic. First of all, does transcranial direct stimulation, does a non-invasive brain stimulation short-circuit the spotlight on important social interventions? Here we're talking about a brain-based intervention, but does that distract us from all of the social causes of crime? Like, maybe we should be targeting poverty, bad neighborhoods. Does that distract from other psychological interventions as well? And similarly, that applies to, fish oil, omega-3 supplements as well. Should we instead be focusing on sort of more, in some ways expensive and effective treatments like cognitive behavioral therapy, for example, more psychological interventions? Should we screen and intervene early in life? It's something Adrian mentioned before. How do we balance the protection of society with, potential stigmatization of individuals if we target them and sort of screen them early in life? Chemical castration, could that be an option for sex offenders? Should they have a choice? And also, the next few questions are sort of more broader questions. Do brain interventions diminish personhood? Does it change the essence of a person? That's an ethical issue that's particularly relevant when we think about these biological interventions. Can prisoners give informed consent for treatment? If we're thinking about these biological interventions. Nor are the days of misusing biological research really over. So we'll leave you with those questions, and that's the end of this talk. 38 minutes.